Welcome to the debate. What if I told you that the perfect healthy gut microbiome, the exact microbial profile you might spend hundreds of dollars on probiotics to try and achieve, could actually make someone on the other side of the world violently ill?
Yeah, I mean, it completely shatters this whole idea that we can just sell a bottled cure for gut health, right?
Exactly.
We are looking at a microbial system that is so hyper-localized, so heavily dictated by where you live and what you eat, that defining a universal, normal baseline might literally be a biological impossibility.
Well, I get that, but that localized variation doesn't mean the underlying rules of human biology have just somehow changed. We're diving into the human gastrointestinal microbiome today, which is this profoundly complex ecosystem that continuously shapes our immunity, our metabolic homeostasis, and, well, our overall physiology.
Right.
So the central disagreement we are exploring today is this. Can a healthy gut microbiome be universally defined, or is the concept of a global standard a biological fallacy driven by radical inter-individual variability? I will be arguing that a universal standard absolutely exists, and it's based on absolute, core, functional, and metabolic signatures that just must be present for a human host to survive and thrive.
And I contend that true ecological health is entirely context-dependent. It's highly personalized and driven by localized evolutionary and geographic adaptations. I think attempting to force a single global standard onto the human population isn't just biologically inaccurate, it's actively misleading clinical diagnostics.
Look, let's get right into the mechanics of this. We need to stop looking at the names of the bacteria and start looking at their job descriptions.
Their job descriptions?
Yes, exactly. I will completely acknowledge that taxonomic variations exist. Like, the specific species of bacteria in my gut probably look very different from yours.
Vastly different, yeah.
Right. But a healthy gut is fundamentally defined by universal non-negotiable functional outputs. There's this mathematical property in ecology called functional redundancy. It basically means that even if taxonomic diversity drops, say because you took antibiotics or changed your diet, the essential metabolic engine of the microbiome has to keep running.
Sure, but
Specifically, I am talking about the continuous biosynthesis of short-chain fatty acids, or SCFAs. Establishing this biochemical baseline is essential for public health policy. We have to stop relying on these subjective lists of, good and bad microbes and just focus on objective metabolic stability.
But those job descriptions, as you put it, are written by the environment. A universal definition of microbiome health is impossible precisely because of the sheer scale of inter-individual variability. I mean, for decades, the medical community operated on the assumption that high taxonomic diversity equated to superior health.
Which we now know isn't always true now.
Right, we now know that is totally false. Take colonic transit times, for example. If a patient has a pathologically slow transit time, meaning food takes days to move through the colon, they often display artificially high microbial diversity.
Yeah, it gets crowded in there.
Exactly. But instead of producing those beneficial fermentation byproducts you just mentioned, that overcrowded diverse community just runs out of carbohydrates, so it starts fermenting proteins, circulating harmful degradation products systemically. Non-genetic factors like geography, diet, and, evolutionary mismatch dictate what constitutes a healthy ecosystem. One individual's perfectly optimized flora is literally another person's dysbiosis.
But, you're pointing to pathological states to disprove a baseline. But the baseline is what allows us to identify the pathology in the first place.
Is it, though?
Yes. If we look past the taxonomy and measure the functional output, the variation starts to look like different means to the exact same end.
Well, the data tells a completely different story. Let's look at the French Milieu Ontario cohort. This was a massive analysis of 858 healthy adults, and it revealed something shocking. Host genetics play a nearly negligible role in shaping the gut microbiome.
Right. It's mostly environmental.
Exactly. Sociodemographics, geographic location, and daily lifestyle accounted for the vast majority of the observable variants. We see this even more starkly in the Helios cohort study in Amsterdam. They looked at over 3,000 individuals across 6 distinct ethnic groups living in the exact same urban area.
Ah, the Helios study.
Yeah, and ethnicity represented the absolute largest source of individual variation. You just cannot separate the function of these microbes from the evolutionary context of the host.
Okay, but you're looking at the wrong variable there. The Helia study didn't just show that taxonomy changed, it proved that the biological engine kept running the exact same way regardless of who was driving.
How so?
Yes, the specific bacterial families differed by ethnicity, but the functional relationships remained perfectly conserved. When specific functional pathways were depleted, it correlated with depressive symptoms across every single ethnic group, regardless of which specific microbes were missing.
So you're saying...
I'm saying the taxonomy changed, but the required functional output dictated the health state.
Well, if the functional output is so conserved, explain the Hadza hunter-gatherers of Tanzania. They consume up to, 150 grams of dietary fiber daily?
Something like that, yeah.
And their gut is incredibly rich in a bacterium called treponema. Now, in a Western industrialized population, treponema is almost universally considered a biomarker of disease. It is associated with inflammation, periodontal issues.
Right, it's a pathogen for us.
But for the Hadza, it's a critical, highly beneficial component of their gut that allows them to efficiently process indigestible wild tubers. Or, look at the Yanomami tribe in the Amazon. Isolated for 11,000 years, they harbor the highest bacterial diversity ever recorded.
And a fascinating resistome.
Exactly. They possess an intrinsic pre-antibiotic resistome. They literally have functional resistance genes to synthetic antibiotics despite never being exposed to them.
Look, the geographic divergence is fascinating, I'll give you that. But think of the microbiome like different engine models.
Engine models?
Yeah. The Hadza are running their engines on wild tubers and baobab fruit. A Westerner is running theirs on a completely different matrix of carbohydrates. Because the raw fuel is different, the parts making up the engine, the bacteria, have to adapt.
Okay.
But despite those taxonomic differences, every single one of those engines is biologically engineered to produce the exact same horsepower.
And how exactly are you defining this universal horsepower?
The active maintenance of a hypoxic or oxygen-deprived environment in the gut. That is the universal metabolic signature.
Right.
Here's how it works mechanically. Your gut is dominated by obligate anaerobes, bacteria that die in the presence of oxygen. Their one universal job is fermenting indigestible fibers into short-chain fatty acids, specifically butyrate.
Which feeds the colon lining.
Exactly. But butyrate isn't just a byproduct. It is the primary physical energy source for your colonocytes, the cells lining your colon. When your colonocytes consume that butyrate to produce energy, the chemical reaction actively burns up any ambient oxygen in the gut.
Creating the hypoxic loop.
Right. It creates a highly specific biochemical feedback loop. By keeping the gut completely devoid of oxygen, you physically suppress the proliferation of facultative anaerobic pathogens, like Enterobacteriaceae.
Because they need oxygen to thrive?
Exactly, and they cause inflammation. That specific biochemical mechanism is a universal requirement for human gastrointestinal health, whether you live in the Amazon, Tanzania, or Tokyo.
Well, you mentioned Tokyo, which perfectly sets up the Japanese sushi factor. This is the ultimate proof that a static global standard is a mirage.
Oh, the parfranase genes?
Yes. Think about this. Japanese populations heavily consume red algae, commonly known as nori, which contains a complex carbohydrate called porphyrin. Human beings do not have the enzymes to break that down.
Nope.
But genomic analyses revealed that the human gut bacterium Bacteroides plebeius actually acquired the genes to break down seaweed directly from ocean-dwelling marine bacteria via horizontal gene transfer.
It's incredible biology.
It is. The bacteria digesting your sushi didn't evolve in the human gut to do that. They literally stole the genetic code from marine bacteria in real time. This adaptation exists in appreciable frequencies exclusively in East Asia and is almost entirely absent in Western populations.
Sure, but
The microbiome is a fluid, evolving entity that rapidly expands its catabolic repertoire to meet highly localized nutritional demands. You mathematically cannot standardize that.
I'm not suggesting we standardize the specific enzymes required to break down seaweed. I mean, that would be absurd. I am arguing that the broader property of ecological stability, maintaining the intestinal barrier, remains a constant requirement.
Okay, let's talk about the barrier.
Let's look at exactly how that barrier functions. In a healthy state, your intestinal mucus layer acts as a physical shield against pathogens.
A critical keystone species here is Akkermansia mucinifera. It makes up about 1 to 5% of a healthy gut microbiota.
Right.
Unlike bacteria that wait for the food you eat, Akkermansia has a unique adaptation. It actively degrades your own host mucin for its energy.
Which, structurally speaking, is a parasitic behavior. It is eating the host's protective lining.
Well, it sounds parasitic, but it operates more like pruning a hedge to make it grow back thicker.
Pruning a hedge.
Yeah. This continuous low-level degradation process chemically stimulates your goblet cells to produce even more mucin, which rapidly restores and thickens the entire mucus layer. Mechanistically, Akkermansia upregulates the expression of tight junction proteins, specifically zonula occludens 1.
ZO1.
Exactly. Think of these proteins like the mortar between the bricks of your intestinal wall. Without that mortar, you get paracellular leakage. Toxins slip into your bloodstream, causing what we colloquially call leaky gut. Beyond that, Akkermansia activates the ALPK1 TIFIR-FAF6, innate immune access.
We need to define what that actually does in the tissue, though.
Sure, it acts like a microscopic alarm system. When it detects the right molecular signals from the microbiome, it actively suppresses chronic inflammation and promotes tissue healing. The presence and activity of these barrier-regulating mechanisms provide irrefutable evidence that specific functional endpoints are globally required.
Look, if this universal standard is so clear and these functional endpoints are so easily defined, why does every attempt to measure it turn into a complete clinical disaster?
Well,
The moment we try to codify your universal functions into standardized markers, we birth a scientifically flawed commercial industry. If you're listening right now and you have ever mailed a stool sample to one of these direct-to-consumer testing companies to find out if your gut is healthy, you really need to hear what the 2026 NIST and Camera Eye investigation just uncovered.
Okay, the commercial direct-to-consumer market is certainly fraught with methodological errors. I'll give you that.
Calling it fraught is incredibly generous. The investigation exposed absolute methodological chaos across the board. They evaluated the analytical performance of the leading microbiome testing services, and the data was entirely incompatible.
The extraction protocols, mostly?
The extraction protocols varied wildly. Some companies had patients ship raw stool in the mail, which leads to massive DNA degradation and completely artificial microbial blooming during transit. The bacteria just multiply in the tube while sitting in a hot mail truck.
Yeah, that's not ideal.
And the sequencing divergence is even worse. You had companies using marker gene amplicon sequencing, specifically the 16S rRNA region, while others used whole metagenome shotgun sequencing.
Which provide entirely different levels of resolution.
Exactly. It's the difference between looking at a blurry Polaroid of a crowd to count heads, that's 16S sequencing, versus doing a high-definition facial recognition scan of everyone in the room. You mathematically cannot compare those two datasets.
right.
Yet companies were using arbitrary bioinformatic cutoffs to tell patients they had dysbiosis. It resulted in one company reporting over 600 species at a low read depth, while another reported fewer than 100 species from the exact same sample.
The bioinformatics were totally unregulated. Yes, I agree.
And because there is no true universal baseline, these companies just arbitrarily invented them. This is exactly why the 2025 International Clinical Consensus Guidelines, which were developed by 69 microbiome experts across 18 countries, explicitly banned the use of phylum-level metrics like the Firmicutes to Bacteroides ratio.
The FB ratio.
Yes, commercial entities were using the FB ratio as a generalized metric to diagnose obesity or inflammation risk. The consensus panel ruled it completely lacks scientific validity because it fails to capture strain-level variations in individual metabolic contexts.
You know, we actually converge entirely on this point.
We do?
Yes. Reducing gut health to simplistic taxonomic lists or arbitrary commercial ratios like the FB ratio is a fundamentally flawed approach. But you're conflating the map with the territory.
How so?
The failure of a commercial test to accurately measure a baseline does not mean the biological baseline doesn't exist. It just means our diagnostic tools are finally catching up to the complexity of the biology.
But if we can't even measure this supposed baseline, what happens when we try to forcefully engineer it? I mean, the failure extends far beyond diagnostic tests. It compromises the very interventions we use.
You mean probiotics?
Yes. Generalized empiric probiotics.
The wellness industry operates on the assumption that if you swallow a pill of universally good bacteria, it will reliably alter your gut flora and repair your system.
Well, we know that mucosal colonization is far more complex than just transit through the lumen.
It is aggressively resistant to universal treatments. Look at the Muspic protocols published by Suez and Alanov's team. They didn't just look at stool samples. They used invasive upper endoscopies and colonoscopies to physically map the highly localized microbiome attached to the mucosal lining of the gut.
Right, a much more accurate picture.
And they decisively proved that just because you find a probiotic in a stool sample, it does not mean it has actively colonized the gut mucosa. Empiric probiotics encounter strict, highly personalized mucosal colonization resistance.
Because of the existing ecosystem.
Exactly. Whether a person's gut accepts or rejects a probiotic strain is dictated by their localized host gene expression and the microbes already living there.
It is highly individualized, yes, which is exactly why precision medicine is taking over.
But it's worse than just being ineffective. Administering these universal probiotics can actually delay healing. When the Suez team examined patients recovering from broad-spectrum antibiotics, the results completely inverted the universal standard model.
The post-antibiotic recovery phase.
Right, giving patients empiric probiotics after antibiotics induced a markedly delayed and persistently incomplete reconstitution of their native microbiome.
Which is counterintuitive.
Extremely. And why? Because the soluble factors secreted by the lactobacillus species inside the probiotic actively inhibited the regrowth of the patient's native, metabolically essential bacteria. Patients who underwent watchful waiting, doing absolutely nothing, recovered faster.
Well, and the FMT patients.
Right, the patients who received an autologous fecal microbiome transplant, meaning they were reinoculated with their own personalized pre-antibiotic stool, showed near-complete recovery within days. Attempting to artificially engineer a healthy state via generalized one-size-fits-all interventions is clinically dangerous.
I hear you. But throwing out the concept of a baseline just because empiric probiotics are a blunt instrument is a huge overcorrection.
Is it?
Yes, the solution isn't to abandon the standard. The solution is shifting our clinical approach toward functional precision nutrition. We don't need everyone to harbor the exact same microbes. We need everyone's specific microbes to be performing the exact same metabolic tasks.
Okay.
And the PREDICT studies show us exactly how this mechanism works in the real world.
The personalized nutrition trials, they certainly highlighted the variability.
They highlighted the variability, sure, but they also proved the functional standard. The PREDICT program didn't just ask people how they felt. They utilized 12 terabytes of metagenomic data.
It was a massive study.
Massive. They strapped continuous glucose monitors to over 1,000 healthy adults and mapped 56,000 postprandial triglyceride profiles, which is just a highly precise way of measuring how much fat and sugar is lingering in your blood after a meal.
Right.
The population coefficient of variation was an astounding 103% for blood triglycerides in response to the exact same plate of food.
I mean, even identical twins exhibited highly divergent glycemic spikes to the exact same nutritional input. Genetics accounted for a negligible 4% of the variability, which reinforces my point. Health is a personalized, emergent property.
But why did they have different spikes? The critical insight from PREDICT is that the functional potential of the gut microbiome was a vastly superior predictor of metabolic health than the patient's genetics or the meal itself.
Because of the microbial clusters.
Exactly. Using advanced sequencing tools like Metaflan4, researchers identified distinct functional clusters of microbial species. The specific microbes dictated how the fats were processed. This is why public health policy must focus on functional diets over taxonomic targets.
Like what, specifically?
Diets rich in diverse, indigestible polysaccharides, like the Planetary Health Diet or the traditional Mexican Milpa diet, universally fuel the essential acetyl-CoA butyrate pathways. We don't need to engineer populations to harbor specific bacterial strains. We just need to provide the universal dietary substrates necessary for those highly localized bacteria to perform the universal job of ecological stability.
Look, you are still trying to force a dynamic, evolving ecosystem into a static conceptual box.
You say we just feed the butyrate pathways, but we've seen that the magnitude and necessity of those functional outputs vary wildly depending on the host's environment.
But the mechanism is the same.
A gut considered perfectly healthy in an agrarian society, dominated by Prevotella to break down massive amounts of fiber, looks entirely different from a healthy gut in an industrialized region. When you attempt to define a universal standard, you risk over-medicalizing normal evolutionary variation.
I'm not trying to medicalize it.
But that's what happens. You risk labeling a perfectly adapted, context-specific microbiome as dysbiotic, simply because it doesn't meet an arbitrary metabolic threshold established by Western researchers. Health is not a fixed biochemical metric. It's an emergent property dictated by the real-time interplay between a highly individualized microbial ecosystem, the host's endogenous physiology, and their specific daily environment.
Well, it seems we are arriving at an incredibly nuanced crossroad here. If I can summarize my position in light of our discussion today, while I fully acknowledge that microbial actors change based on geography, ethnicity, and lifestyle, the biological script they are reading from does not.
The script?
Yes. Ecological stability, active barrier maintenance through mechanisms like Akkermansia muciniphala pruning the mucus layer, and the consistent biosynthesis of short-chain fatty acids remain a universal mandate for human health. The engine parts may look different, but the horsepower required to keep the human machine running is an absolute, quantifiable reality.
And, well, my summary is that the sheer complexity of the human host overrides the desire for a neat, universal definition. The catastrophic failure of commercial dysbiosis tests, the active harm caused by universal empiric probiotics, post-antibiotics, and incredible localized adaptations, like bacteria stealing genetic code to digest sushi, all point to one truth: health is contextual. The idea of a global standard ignores the incredible evolutionary plasticity of the microbiome.
Well, we certainly agree on one major front. Relying on blurry diagnostic Polaroids like 16S sequencing or simplistic commercial metrics like the Firmicutes to Bacteroides ratio is a deeply flawed approach that belongs in the past.
Absolutely. We converge completely on the necessity of demanding rigorous, high-resolution functional data. Where we continue to disagree is whether those conserved metabolic functions constitute a true global standard, or whether they simply reflect parallel, localized adaptations that fluctuate far too wildly to ever be standardized.
Which means there is tremendous value in holding both perspectives simultaneously, appreciating the strict biochemical functional requirements of the human gut while simultaneously respecting its incredible evolutionary plasticity is essential for the future of precision gastroenterology.
There is vastly more to explore in this material. I mean, we're only just beginning to decipher the real-time dialogue between our human cells and the trillions of microbial passengers we carry.
It brings us right back to our opening thought. Whether your microbiome is perfectly adapted to digest wild tubers in Tanzania or seaweed in Tokyo, assuming we can force every human being into a single commercial definition of health might be the biggest blind spot in modern medicine. Thank you for joining us on The Debate. Keep questioning the map, and we will see you next time.